Okay, so good morning everyone. So I'm Keat Lay. I'm the field application specialist from First Base under Epica. Okay, welcome to our global webinar with the title Mastering Sample Preparation, Achieving High Purity and Yield from Soil and Stool Sample for NGS application. Okay, so upon the whole presentation, if you have any questions, do type into the chat box. So I will try my best to answer you after the presentation, after the sharing. Okay, and there may be some of the question that I cannot answer you on spot, so maybe need more information or what. So you may email me at support@epicalscientific.com, so we can discuss from there. Okay. Yeah. So not to delay, let's start the presentation now. Okay, so for today, we are going to share with you some tips to help you in mastering sample preparation, typically on soil and stool sample for NGS application. And I will go through a short introduction on the quality of the DNA that is needed by NGS application. Then we'll go into detail on stool and gut DNA extraction in part 2, and then soy DNA extraction in part 3. Okay, so there'll be 3 parts in this sharing. Okay, so for the first part, we need to know what is the requirement for NGS sample. NGS sample requirement are usually specific according to different application in order to ensure the result accuracy. So it means different application with different platform will have different sample requirement. Okay, so we categorize all these requirement as NGS grade sample requirement. Now, what is the difference of this NGS grade sample with the normal standard grade? Commonly, we will categorize the extracted DNA into these 2 grades just to ease the communication. So the standard grade DNA usually refer to DNA used in some basic molecular application like PCR, cloning, or routine qPCR or fragment analysis. Usually for this application, the quality level of the DNA is not so strict. So you may have some impurity in your sample. You may have degradation in your DNA, and they are okay to be used. Also, the amount of DNA needed per reaction is usually not very high. It may be like 10 to 20 nanogram per reaction. So it means the yield of the extracted DNA no need to be too high, and even there are contaminants or possible quality of the DNA may slightly different between the sample, but they do not really affect the result. So you may also can have a basic quality control to check the concentration and purity with spectrophotometer. Then the DNA will be good to be used for basic molecular application. But in NGS grade DNA, they are usually restricted to follow the requirement of the application and the platform used. For example, like whole genome sequencing, then you may need intact DNA with high molecular weight. Smearing may not acceptable. Okay, but in empirical sequencing, some degree of smearing is acceptable. Okay, so it depends on the application. And NGS grade DNA, they will need to free from inhibitor or have very low impurity in the DNA, because this contaminant, they will affect the library preparation, which will result in inaccuracy in the NGS result. Also, the DNA amount need to reach the requirement, which usually is higher when compared to standard grade DNA. It may be like 50 nanogram per sample, or some may go up to 200 nanogram per sample. Okay, so for integrity of the DNA, it may also depending on the platform used. So, for example, if the platform is for short read like Illumina, then you may have some degree of the degradation of in the DNA is still acceptable. But long read platform like PacBio or Nanopore, then it may require higher quality of DNA with good molecular weight. Okay. Okay. Another important point for NGS grade DNA is the extracted DNA will need to be highly reproducible. So to ensure the reliable detection, especially on mutation or biomarker study across multiple or different samples. Okay, so a more comprehensive QC will need to be done for NGS sample. At least you will need to run a gel to check for the DNA integrity and also determine the purity with spectrophotometer. Okay, so both of it is a must. Okay, yeah. So here, I just would like to share a little bit on the quantification of DNA. A comprehensive QC of the DNA usually will involve checking for DNA profile, purity, and yield of the DNA. The DNA profile is to know whether the DNA is high in molecular weight and is there any presence of like contaminant like RNA or protein. Okay, and you can use agarose gel electrophoresis for this purpose. While for purity and yield, you can determine them through spectrophotometer or nanodrop or qubit. And in the next few slides, I will share some tips on what you should expect for your NGS grade DNA and what you do not want and how to overcome them. Okay? Okay. For DNA profile, check using the common one is the agarose gel electrophoresis. So when quantify the extracted DNA using the agarose gel electrophoresis, you should expect an intact band with high molecular weight, which usually show you a single band above the DNA marker because genomic DNA have a huge size. Okay, and this band should be sharp with no smearing around it, and also there will be no RNA found at the bottom of the gel and no protein contaminant, which it will causing the DNA to stuck in the well. Okay, so this is what you should expect for your the best DNA. Okay, if the DNA has a different degree of degradation, then you'll find smearing showing in your gel. It can be a total smearing like this, or maybe there is some intact in the gel and you can still find some smearing also. Okay, because due to different degree of degradation, and this usually caused by enzymatic activity like DNAs in the sample, or maybe oxidation damage or UV radiation or hydrolysis. Okay, so DNA degradation happen from the minute after sampling a specific a specimen, and it continue to degrade regardless of how the DNA have been preserved. It's either you can slow it down or by preserve it, or you can have a better one is to extract the DNA immediately. Okay, yeah. So Within 24 hours, if you're going to extract your DNA, so you should store your sample under a sub-zero temperature. But I understand that sometimes from the sampling field to the lab, it may takes you more than 24 hours, or due to the field condition, it may causing the sample to be stored in a not a very well optimum condition. So in that case, then you may need to preserve your sample with some preservation solution. Okay, so the best preservation protocol is highly depending on the organism, on the sample type, on the study design. Okay, for example, you may use the DMSO, it may suitable for mammalian tissue, okay, something like that. Okay, and you should actually test a few methods of storage and preservation of the sample, included the factor on possible field condition, the cost, the time before you go to sampling in the field. Okay, and finally, after the DNA extraction, do store the extracted DNA in a solution that buffer the pH, and usually it will be the DNA elution buffer which carry trace. Okay, yeah. So if the DNA is contaminated with protein, it can actually alter the DNA migrate in the gel and cause the DNA to remain or stuck in the well here. Okay, certainly DNA stuck at the well possible also due to some incomplete well formation, and that is why you should always have a DNA marker, so and also the positive control to run together with your sample in a gel. So from here you know, okay, the gel is well formed. Okay. Okay, so what is the cause for protein contamination? It can be due to too much of the sample used in the DNA extraction, which causing the protein was not completely removed or dissolved. Okay, it can also due to the proteinase K, it do not work in the DNA precipitation step upon your DNA extraction. So usually, what used to degrade protein is the proteinase enzyme, and proteinase K is the most commonly found in all commercial kit for DNA extraction. So you need to store the proteinase K properly in order to ensure its activity. Proteinase K, it should store at minus 20 degree if it is in lyophilized format, and in that case you can store it up to 2 years, or in solution then you can store it up to 1 year. Okay, so after the DNA extraction, and if you found that there is still present of some protein in your sample, you can also perform a phenol ethanol precipitation just to remove the protein. Okay, yeah. Okay, next is for RNA contaminated sample. So you will find that some there will be some smearing at the bottom of the gel, is the cause for RNA contamination, and usually it is because also due to too much of sample used in the DNA extraction. So RNA, it could comprise 60 to 80 of the nucleic acid that is present in the sample. So if too much of the sample used, it may causing incomplete removal of the RNA from the sample. Also, there is possible that the RNase enzyme used in the DNA extraction kit is not working well, or the activity may be reduced. So what you should do to avoid RNA contamination in your DNA sample is you should make sure to choose the DNA extraction kit that include the RNase degradation step. And commonly you will find RNase A in most of the commercial DNA extraction kit, and it helps to remove RNA during the DNA extraction procedure. So usually this enzyme is kind of stable, so you can just store it at room temperature for up to 6 months, but still the best to store it at -20 so that you retain its activity. Okay, yeah. Anyway, you still can also have a post treatment to remove RNA with the RNase enzyme later on after DNA extraction. Okay. Okay, so after the DNA profile with agarose gel electrophoresis, quantification of the DNA with nanodrop or cubic is also important. Usually, we recommend to use nanodrop rather than cubic, because nanodrop is able to give you the purity check at adsorbent 260-280 and 260-230 ratio, and also the concentration. Okay, but cubic it only enable you to check on the concentration. Excuse me. So if you are using nanodrop, you should expect to get the adsorbent ratio at 260-280 at 1.8 ratio, okay, reading. So this indicate pure DNA. Lower of this reading, it may tell you the possibility of contaminant by RNA or phenol in the DNA sample, or contaminant by protein in the RNA sample. Okay, higher of the reading is okay, while for adsorbent at ratio 260-230, you should get around 2 for pure DNA. If the reading is lower, it means there is contamination of salt, sugar, or other organic compound. While higher, it means the blank is incorrect, so you need to use the correct blank. Okay, but nanodrop is a spectrophotometer, so which it using light absorbent to detect what is inside the tube, inside the sample. And because of this, it may not give you a very accurate reading for the DNA concentration in your sample, especially when your sample is low in concentration. So if you need to get a more accurate number, then you can use cubic. Okay, cubic is using a fluorescent based quantification method, so it can give you an accurate reading if the DNA concentration is low, but it do not able to be used for purity check. Okay, yeah. So why a high purity DNA and yield is important for NGS grade? Why you need to perform all the QC just now, just to make sure your sample is NGS grade? Okay, reason is, okay, purity. Purity, it is important in determining the accuracy in the sequencing result, because if there is impurity of presence of inhibitor in the sample, for example, humic acid from soil or some bile salt from stool sample, they can interfere the DNA polymerase or reverse transcriptase used in the NGS library prep or PCR enrichment. Okay, so this, it can lead to misreading or incomplete sequence generated, or some background noise that can compromise the accuracy of the sequencing result. For example, low purity DNA. It may cause uneven base calling and result in incorrect variant detection. Okay, the purity of DNA will also determine how good your library prep can be. So, as we know, library prep in NGS may require enzymatic steps like PCR, like fragmentation, like and repair or adapter ligation. Okay, so if there is inhibitor present, they can reduce the efficiency of all these enzymes and leading to incomplete library. So poor library quality, certainly it will result in low read depth and also uneven amplification across samples. And also it may introduce bias in the sequencing data. So using a high purity DNA, it can help you to make sure the process is proper and ensure the consistent library preparation is generated. Okay, and also contaminated DNA, it can also cause instrument error or poor cluster formation on the platform, for example, like in Illumina, and leading to incomplete run. Okay, so fear of this, it may cause you the time, the cost, the risk of sample degradation. So try, we want to reduce all these possibilities. So you need to make sure the purity of the sample is good. Okay, as for the yield, why yield is important for NGS application? Many NGS application like whole genome sequencing and metagenomic, it require at least 50 to 20 nanogram of DNA per sample to generate a sufficient library. So insufficient of the DNA, it can result in poor library complexity, limit the coverage, and also it will maybe need to have multiple amplification steps to reach the target amount. So because of multiple amplification step, then it may reintroduce bias in the sequence. Okay, so with good high DNA yield, you can ensure a smooth downstream workflow with the minimum amplification. So this can also preserve the nature of the sample. Okay, so sufficient yield can also ensure the consistent sequencing depth across all the region of the genome or amplicon pool, and preventing a gap in the coverage. Okay, yeah. So from all the previous slide, we know that NGS grade DNA that we should expect from our DNA extraction. Okay, and we know it's important because it will affect your result. Now we go into the detail on the DNA extraction from difficult sample. Okay, the first one we will share is on the stool and gut sample. Stool and gut sample, they are difficult to extract, but they do have high impact in research. Okay, there are a lot of study using stool and gut sample, and all these study do have high impact to us. For example, the study on stool and gut sample on the pathogen related to diarrhea or related to COVID. Okay, also on autism. Okay, and this sample will also use in a study for for us to know the behavior of animal in a region. Okay, so the accuracy from the NGS result are very important as they give impact to the research and even to our daily life. For example, on the disease study. Okay, so but we all know DNA, to extract DNA from stool and gut sample is difficult. Okay, why? Because the nature of this sample, stool or gut sample, they contain complex composition. For example, the whole cell, also some food particle that have complex polysaccharide inside the food. Okay, there are also present of a lot of inhibitor in this sample. For example, the bile salt, phenolic compound, or other organic compound that are difficult to remove upon DNA extraction. And stool or gut sample, they carry diverse of microbial in their samples, and this microbial diversity in the sample, it it can make it difficult to efficiently lyse all the cell, or to recover their DNA upon DNA extraction. And sometimes the condition of the sample itself make it difficult to extract. For example, the stool may be a hard or dry stool, or stool from herbivore which have a high in fiber. Okay, or maybe a sample from bee guts or insect guts, where the DNA abundancy is very less, then it will be difficult for you to determine the DNA. So you need the optimized protocol for this. Okay, for all this, they may affect the DNA extraction. So the challenges that commonly found by researcher when extract DNA from stool and gut sample are um, the variable of DNA quality and quantity. The quality and quantity of the DNA determined from different batches of extraction always found to be vary most of the time, because it depends on the diet, it depends on the health status of the individual, and the stool consistency. And this variability, it can make it difficult to standardize extraction protocol and compare the result across the study. Okay. Also, second one is there is possible of contamination. um, Contamination can easily found in the DNA extracted from stool and gut sample. Not only the organic compound that mentioned just now that is inside the sample, but it can also come from external source. For example, elephant stool that may collect from forest, it can carry the DNA from other animal also. Okay, so it may be difficult to ensure the purity of the extracted DNA. Then the stool or gut sample, they are full of enzymes, especially gut sample, which usually contain high enzyme, such as nuclease that can degrade DNA. And if this enzyme did not remove, they will cause degradation and affect the quality of the DNA extracted. And because of all the above, optimization on the extraction protocol for stool and gut sample can be very time consuming, and it require a significant optimization to maximize the DNA yield and quality of the extracted DNA. So all this mentioned, they can affect the downstream application like empirical sequencing and directly influence the data accuracy and species abundancy in the result. So how to overcome this? One of the way is to choose for a suitable DNA extraction kit. And yeah, allow me to introduce our primary stool DNA extraction kit, which can help you to resolve all the mentioned challenge in the previous slide. So how this kit can help you? Primary stool DNA extraction kit, it has a high versatility. It is suitable for to be used for human and animal stool extraction as well as gut content. And it works well for normal stool, for dry, hard and high fiber stool also. And with the combination of ceramic bit harmonization and chemical lysis method, primary stool DNA extraction kit can give you a high yield, high purity, inhibitor-free NGS grade DNA. Okay, and the extracted DNA proven to be suitable for various type of application, include PCR, include Southern blot, Amplicon seq, and also qPCR. Okay, just an insight of the primary kit. So how it can help, you can find that there is specific protocol so designed to tackle different type of sample. So to it help you to determine a good yield and good coverage rate for the extracted DNA. So also it means there is less optimization that you need to do. Okay, you just need to refer to the protocol, and you will always found there are some notes in our kit manual. Okay, because our lab, as mentioned by Mr. Dino just now, our lab have tested many different type of samples, and we know that what usually need to be take care by the end user in order to help you to determine a NGS grade DNA. Okay. And as mentioned previously, stool sample, they carrying high amount of inhibitor. So to help you in getting good quality DNA that is free from inhibitor, we have implement the inhibitor removal step in our primary kit. So the protocol, so you just need 2 minutes, then it can help you to get rid the inhibitor through this simple step. Okay, and in case you need a DNA with high purity and without a trace of RNA, you can also follow the RNase treatment step here. So it is just a one step, then it can help you to get rid of the RNA contamination in your sample. So you no need to go and find any like post treatment with the RNAs for to get rid of the RNA. Okay, so it's a fast and easy method. Okay, so here, just to share the performance of primary stool DNA extraction kit that we have in our lab. So this is a data from some of the sample that we have tested. Okay, so here we have extract DNA from 100 milligram of animal stool, and from cat, dog, horse, and mouse gut. Okay, and then we have also extract 200 milligram from human stool. So the graph here, it show the DNA yield and purity. And you can see that DNA extracted by primary stool DNA extraction kit, it give you a high yield and high purity. Okay, so however, as mentioned along this presentation, the nature of the stool sample, they are varying. So you can see from the gel photo here, human stool it still can give you a good intact band. Okay, but horse sample, it will give you degraded DNA. Even though we compare the extraction with other two brands also, the sample quality from this horse sample is also the same. So this proven that it is the nature of the stool sample that affect the DNA. Okay, yeah. And we have tested the extracted DNA by amplified with primer for 16S V4 region, and we found that the amplification are good. So all the extracted DNA, they are free from PCR inhibitor. Okay, so it means they are good for library preparation in the downstream application. Okay. So also we proceed the DNA extracted from human stool from different commercial kit and also primate to amplicon sequencing by amplified with the primer targeting 16S V4 region and sequence using Illumina MySeq platform. So after that, the data were analyzed using the SHIME tool pipeline, and here is the comparison result for 16S microbial profile for the DNA extracted. Okay, so from here you can see that the microbial profile generated from three kit are comparable and with about the similar species abundance. And meanwhile, you can see that primate, it has highest in the species richness. And also you can see there is gram positive and gram negative bacteria here. So it means there is no bias in the DNA extraction by using Primary's 2 DNA extraction kit also. So this proven that the extracted DNA is good for NGS application. Okay. Okay. And also because we know stool sample collection, storage, and transportation may be troublesome as it need to keep the sample in -80 to maintain the DNA inside the sample. So here we have additional um, stool DNA extraction kit, which can help you in all this. Okay, so there are a lot of different kind of stool collection kit that you can use. But usually after collection, you will need to store the stool sample at least 2 to 8 degrees Celsius upon transportation. And if you're going to extract it in the same day, so if you're going, if the DNA extraction is not going to perform in the same day, then you need to store the stool sample in -80 until processing. Okay, and this may be may make it difficult for you to transport the sample or also store the sample if you have a big amount of sample for your study. So to ease the process of storage and transportation of the stool sample, and in the meanwhile, preserve the DNA, we designed the stool collection kit here. Okay, so it is a tube that is designed for collecting, preserve, and storage of the stool sample, typically for DNA extraction. Okay, and the tube, it has a integrated spoon, which it is the collection process. So what you need to do is just to transfer 2.5 gram of the stool sample inside this tube and shake vigorously. Then the collected sample inside this tube, it is good to store at room temperature. And as the tube contain a non-lysis solution, so it ensures the stability of the DNA in the sample. So you can also transport and store the sample in room temperature for at least up to 2 years. Okay, and we test the sample performance. So we extract the DNA collected and store at -80, and also sample collect and store at our Stool DNA collection kit, and also compare with a stool collection solution from a brand. Okay, and from here you can see that the microbial profile for first base stool DNA um, collection kit, and also from brand Z, um, they are quite comparable with quite similar species abundancy and the species richness, okay, from all the extracted DNA. And so it means our first base stool DNA collection kit is working well in preserve the DNA even though it is stored at room temperature only. Okay, so yeah, we go into part 3 of the sharing. So here is another difficult sample. We are going to discuss on the DNA extraction for from soil sample. Okay, there are many study on soil sample which also have will bring impact to us. For example, analyzing the soil sample to understand the microbial community structure and function for agriculture study, or using a metagenomic study on soil sample to determine the information for forensic investigation. Also, disease study using NGS method on soil sample to know the geographic distribution of a disease, okay, or a transmission of the disease through the environment like soil or water sample. Okay, so we all know that extracting DNA from environmental sample, because they will be difficult, for example like from soil or sample, because soil from one place to another it's different, okay, and water also. Okay, so what can be the challenge that is determined in NGS grade DNA extraction from this soil sample? The first is there can be presence of inhibitor in the soil sample. So this is usually the main challenge. There are present of many different type of organic compounds like polysaccharide and protein that may result in contamination and reduce the purity of the DNA extracted, and which it subsequently will interfere the downstream application. Okay, another challenge is the low DNA yield in some of the soil like clay. Okay, which it contain relatively low microbial biomass due to the poor in nutrient or oxygen, and it may be also a sterile environment. So this means the total amount of DNA available in this soil is limited, and so the extracted DNA may be insufficient for your NGS workflow. Okay, and DNA degradation can also complicate the DNA extraction process. Okay, so microbial DNA is proven to break down by nucleus and may also be damaged by temperature fluctuation and the microbial activity over the time. So this lead to short or degraded DNA that is difficult to amplify or sequence later. Okay, and next is the heterogeneous nature of the soil. It may make the DNA extraction complicated. Soil is made up of different particles, including clay, including sand, and it can bind or trap the DNA and reduce the efficiency of lysis and extraction later on. And additionally, you can have co-extraction of some non-DNA component. It can be like the humic acid or fulvic acid that can bind to the DNA or interfere the enzymatic reaction, such as PCR and NGS library preparation later on. And additionally, metal ion that like uh, iron or aluminum that present in a soil sample can inhibit DNA polymerase, and it will interrupt the downstream application. Okay, and lastly is the microbial diversity and composition of the soil itself can also be a challenges. Okay, especially in lysis of DNA extraction. So because different microbial species, they have different to respond to lysis. For example, you have gram positive and gram negative bacteria. So they have different response to lysis. Okay, and this can lead to bias in DNA extraction and causing incomplete recovery of the microbial community in the DNA profile. Okay, all these challenges, it can affect the accuracy of the NGS application later on. So you need to have an optimized protocol to tackle all these challenges. Okay. So please allow me to introduce the primary soil DNA extraction kit. So this kit, it is designed with strong lysis buffer and inhibitor removal step to ensure the determination of NGS grade DNA for your downstream application. Okay, so primary soil DNA extraction kit. It is versatile and it can work for all three types of environmental sample, which are soil, manure, and water. Okay, the kit is high in efficiency and it has the combination of both chemical and mechanical lysis method to ensure complete lysis of the sample. Okay, because the DNA in some of this sample may be low in abundancy, or some of them may have high contaminant. So it is important to have an optimized protocol to lyse all this sample. And the kit, it is also effectively remove abundance of humic substance and pigment. So ensure the DNA extracted is high in purity and integrity, which are suitable for a wide range of application like PCR, certain blot, enzyme digestion, and also NGS, typically amplicon sequencing. Okay, so you can see that in primary soil DNA extraction kit, we have include a very comprehensive protocol from sample preparation until lysis of the sample up to DNA elution in order to ensure the whole extracted DNA is suitable for your NGS application. And the protocol, they are customized for specific sample type to make sure the determination of high DNA yield. Okay, also you can see that we have a special buffer, SIR buffer. It is help to clean away the pigment or inhibitor like humic acid, typically in soil sample. So it is just one step here and it can help you to remove the inhibitor. Okay, so the handling process is very easy. Okay, And here this just to share with you, this is how our soil bead tubes looks like. So it carry the glass bead for mechanical lysis of the soil sample. Okay, for our SIR buffer, it is effective in remove pigment or other inhibitors, but in the same time, it may reduce some of the DNA yield. So, as you can see here, we have the data share here in the table. Okay, so for the DNA extraction with and without the SIR buffer, but we do understand that in some of the soil sample, like clay, the DNA amount is expected to be low. Okay, so maybe this SIR buffer may not suitable to be used. So we will advise the user to skip this step of the SIR buffer here, okay, and go direct into the next step. So if after the DNA extraction, you find that there are present of impurity or pigment in the sample, you can we have include another protocol to help you in clean up the DNA. Okay, yeah, it is the support protocol which help you in the DNA clean up here. Okay, yeah. So, using the DNA cleanup protocol, uh, we it is proof that it can remove the pigment, okay, and also the inhibitor like humic acid, okay. So in the DNA, and it can give you high recovery, more than 85% of the DNA in the sample. All the protocols in primary soil DNA extraction kit was optimized and tested in our lab for the performance. Okay, so here is some of the soil sample which used in the performance test. Okay, so we have clay, which is a good representative for low DNA abundancy soil sample. Then we have also organic soil and that is high in microbial diversity. Okay, also we have forest soil and also some soil from agriculture. Okay, like the soil from paddy field, oil palm estate, vegetable bed soil for the test. Okay, so let's see what is the performance. So we extract 500 milligram of the soil from each of the sample mentioned just now, and it proved that primary kit works well for various type of soil sample, and it able to give you DNA with good purity at absorbance ratio of 260-280 of at 1.8 to 2, and you can get good yield from this sample. The extracted DNA showing intact band, okay, with low smearing in the agarose gel. Okay, so we proceed the DNA to PCR with 16S V3 V4 primer and also ITS region uh, to test the inhibitor, and we found that all the extracted DNA they are good in giving you amplification. So this means that all the extracted DNA is free from inhibitor and can proceed to PCR QC in especially in empirical sequencing. And like you saw just now, okay, the DNA amount for the clay sample, it has the lowest DNA abundancy. Okay, so it give you a low yield. So here we extract DNA from 200 milligram of clay soil, following the protocol suggested by each kit. So we have a kit comparison between primary with brand M, brand PL, and Q here. Okay, so you can see that primary and brand M, it can give you some reading. Okay, and okay, because here the initial extraction is using 200 milligram of the soil sample only. So we did increase the use of the sample to 500 milligram, and we do determine some reading for brand Q also. So we able to proceed the all this sample from primary, brand M, and brand Q to NGS amplicon sequencing. Okay, and here is the data comparison. So from here you can see that the microbial profile generated from all these three kits are comparable in species abundancy and also species richness. Okay, and there are no bias observed, okay, in the DNA extracted from this kit, as you can still able to find the gram negative and gram positive bacteria in the phylums here, top 10 phylums here. Okay, so it means that the DNA extracted from primate kit is proven to be suitable to be used in NGS application. Okay, yeah, so here come to near end of my sharing. As a summary for this whole presentation, so in the first part we of the presentation, we have explored the concept of NGS grade DNA, and we understand how DNA purity and yield can direct impact the quality and accuracy of your NGS result. Okay. Also, we have covered the essential technique to for you to quantify your DNA to ensure they meet the NGS grade standard. Okay. And then in the second part, okay, and the third part of the presentation, we discussed the two very challenging sample type for DNA extraction, which are the stool and soil sample. And in part two, we focus on the stool sample, where we go through the nature of stool sample and the challenge that is in extracting DNA from this stool sample. And similarly, in part three, we also dive into the challenge faced by a researcher in DNA extraction from soil sample. Okay. And along the side of this discussion, we have introduced the primary stool and DNA extraction kit, and also primary soil DNA extraction kit, and highlighting how this kit simplify the DNA extraction process for the challenging samples and ensure the reliable and high quality result for your NGS. Okay, so private DNA extraction kit, it can provide you with DNA of high purity and yield, specifically meeting the stringent requirement of NGS application, as we have also show you the proof under our performance test. And this ensure the reliable and high quality result in application that demand accuracy and sensitivity. Okay. Also, with the streamlined protocol, primary kit makes the DNA extraction process very straightforward and efficient. So the customized workflow, it can minimize handling error and also making it accessible even for those new in complex sample type like soil and stool DNA extraction. Okay. Also, with the performance comparable to establish brand in the market, you can see that Primeway, it can actually deliver excellent value by providing you a high quality DNA at a better cost. And you can even use it for your basic molecular application, especially for PCR or single sequencing, if you are working on stool and soil sample. Okay, yeah. So that's come to the end of my sharing.